Good evening, everyone. Very warm welcome to all our participants and our speaker. Hope you all had a great day. Myself, Minoti, I will be host for the day. Today, we will be conducting our 30th session of Student General Club organized by BioPractify and partners. Topic for the session is Microbes for Future. This will provide an in-depth look at how these tiny yet powerful organisms play essential role in our daily lives. The environment faces many challenges, and one such major problem is impact of microplastics. Microplastics which persist in the environment and their contribution to pollution urgently demand sustainable alternatives. One promising solution lies in utilizing microorganisms. And Today's speaker we have is Ms. Shruti Kutmutia, and she's a leading expert in this field and will offer you clear insights into how these processes work and the innovative solutions being developed to address them. Without a further ado, I would like to give a brief introduction about her. Ms. Shruti Kutmutia is a bioinformatician and an applied biotechnologist with an impressive 8-year research career at NTU, Singapore, where she specialized in aerial metagenomics and global microbiomes. She is a visionary founder of BacAlt BioSciences, that is Bacterial Alternatives, a company dedicated to creating high value microbial biofilms from agro industrial waste to replace microplastics from PCCPs and agricultural product formulation. It is also paving the way for future where microbes are at the heart of sustainable innovation. Welcome, ma'am. We are
Thank you.
incredibly grateful to have you here, and I would like you to take over and start with the session.
Awesome. Thank you for giving me the opportunity to talk about my work and about microbes. Microbes are literally my favorite topic to talk about. I started my journey or interaction with microbes in bachelor's. I did my bachelor's in microbiology from Pune, Ferguson College. Never looked back actually. After that, microbes have been everything. Later, after studying microbiology, excuse me, I got interested in the genetic features of it as to why do microbes behave the way they do. If you talk about the evolutionary theory, or if you talk about theories of the oxygen catastrophe, where the world did not have oxygen before microbes existed, and then cyanobacteria existed, which started spitting out oxygen in the world, and the entire landscape of the world changed. Earth in itself changed, and oxygen actually started being in the atmosphere through bacteria. Those are some kind of facts that make you wonder, like, what are these invisible creatures and how can they do what they can do? So then I did my master's in bioinformatics and applied biotechnology from uh, IBAB, Institute of Bioinformatics and Applied Biotechnology, which is where I'm housed currently for my startup. Here, when I was studying, 2016 to 18, metagenomics was quite new. It was not something that was well taught or even perceived. And I convinced my professors here to teach me a course there, and they did. They were kind enough to just whip up a course and teach me metagenomics. And then I developed an even bigger interest in how can you engineer um, environments. How can you do life science engineering by engineering microbes or controlling the community of microbes? What do they tell you? How can you learn from it? That's where I chose to go to Singapore Center for Life Science and Engineering, CELSI. It's the world's leading biofilm research institute. What they do is essentially, one second, sorry. Essentially, what happens in CELSI is air, water, skin, all kinds of biofilms are researched to understand that microbes are not, they don't act alone. They act in communities and they interact with each other quite a lot. That is through biofilms. Now, this is a much more advanced technology or much more advanced way of looking at microbes, not as an individual microbe, but as a colony or as an organoid, like an entity. So if you take, say, a Petri plate, the microbe in the one corner of a Petri plate is communicating with the microbe on the other corner of the Petri plate, acting like an organ or an entire community. So that's the kind of research that is explored in CELSI, mostly through genomics route. Our lab used to completely work on aerial genomics. And I'm just going to walk you through a presentation of a quick short review of what I've done in the past and what is my current company doing. And let's have an interactive session. I'm happy to answer whatever questions you have as we go. It'll be nicer like that. One second. Do let me know if you can see my screen. Can you guys see my screen?
Yes, it is visible.
Awesome. And now,
Yes, we can see your screen.
awesome. So I am the co-founder of BacAlt BioSciences. BacAlt stands for Bacterial Alternatives. What we do is we manufacture different kinds of products that can replace microplastic generating plastics or PMMAs, nylons, Teflons in ingredients. The problem statement came uh, when we went through the ingredient list of products, right? And then when you see that, oh, conditioners, shampoos, mascaras, lip care, everything has plastics in the ingredients. It is a known fact that sometimes the formulation itself has more plastics than packaging, which is completely lost on consumers, right? So that's that one problem that really bothered us, me and my co-founder. And then we realized that okay, there's a scope here, and let's start this company and see what we want to do. So we have developed three patentable processes for manufacturing of bacterial nanocellulose and polygamma glutamic acid. Pranav and I have been friends since 2012. We did our bachelor's together in Ferguson in microbiology. I told you my background, but Pranav then further went on to do his PhD from NCL Pune. He holds multiple global patents, multiple Indian patents, very well published, and is really, really an expert in fermentation processes. So I'm just going to give you a snapshot of my previous 8 year work. What we did was we understood the air microbiome of the world. We have 33 countries sampled, 104 sampling locations. We found innumerable bacteria and fungi. And in our upcoming paper, what we do is we tell you, is it different? Do you see difference in the air you breathe in India versus Indonesia versus US versus Japan? And yeah, this was my team back in the day. We still interact quite closely, and that's Professor Stefan Schuster. He's the most vision-driven guy I've ever met in my life. If you're interested in metagenomics or aerial microbiology, he's your guy. He is a revolutionary guy in his field. Then coming back to what we do here at BacAlt. Yeah, sure. We know it's a big problem. It's a $66 billion opportunity because plastics not only go in face washes and shampoos, they also go in toothbrushes, so oral care. They also go as binders in medicines, so nutraceuticals or pharma. They also go in agriculture for water holding capacity. So there's agri too, right? So how do we find a solution that can satisfy all these ranges? Then we thought of biopolymers in the aspect that how do we make it platform biopolymers. Then we went to the older books of polymers. We saw that, oh, there's PHA, PHBs, PLAs. You must have heard a lot of these biopolymers coming in, right? More often than not, they do make phthalates. So you must have seen that there are some bioplastics that are only degraded in water, some bioplastics that are degraded under soil, under this temperature, under that temperature. So there are a lot of checkpoints that you have to crack when it comes to biomanufacturing. It already in itself is really difficult because the yields, right? The yields of mining or petrochemical can never be similar to the yields of making a bacteria produce it. So we had to come do an entire thesis search of what polymers we want to work with, and we ended up with 200-year-old biopolymers, bacterial nanocellulose, and PGF. One is a polysaccharide and one is a polyamine. What polysaccharide does give you is a free OH group. And what polyamine gives you, especially PGA, it gives you a free COOH group. So there's an acid group and there's an hydroxyl group. Now, if you talk about chemistry in that aspect, these two groups are very essential to interact with water or to interact and make derivatives out of the product. So when you make this available, green chemistry becomes quite easy and well known. One of the reasons to select our biopolymers was that. Another reason is it is pre-approved by US FDA. These polymers are edible. They are compatible, very compatible with the skin. You can put it on open wound, no problem. And they have their own journey of getting the approvals. But today, if I have to sell it to pharma, I can without having any certification. So that reduces our entry to market. And yeah, that's why we started with the product. Another in-depth information for our let's talk about cosmetics, right? When it comes to L'Oreal, oh, these are the most common brands that are available in India right now. Um, they also have a range of products. You can buy a L'Oreal shampoo for 500 rupees, but you can also buy it for 2000 rupees. So more often than not, the 2000 rupees shampoo, sorry, 500 rupees shampoo is going to be mostly plastics. Now, plastic is a design flaw to begin with, but it also adds some features like bulking agents, film formation, emulsifier, glitter, exfoliants, etc. So when we talk about micro beads or the flakes in the toothpaste that you see, they are a design element, but they can also add functionality. Plastic is a necessary evil, right? It got its fame because it's completely malleable. The petrochemical can be transparent, translucent, water soluble, water insoluble, liquid, solid, blue color, purple color, anything. That's one reason why plastic can be used in so many areas, right? So we also want to make and choose the polymers that can replace plastics one to one when it comes to not just functionality, but also being very, very malleable. Uh, just a trivia, India spits around 1.8 billion grams of microplastics in the sea just by using the toothpaste available. You must have just recently read the news where everyone said there was a research, some study done that every Indian sugar and salt brand has plastics. Now, if you're spitting 1.8 billion grams of plastics in the soil through the sink, these products don't have an option of recycling because they're already in the microform. You cannot recycle anything in the microform yet. So the only option there is replacement. Another thing that happened, so India has no data of the effect of such plastics in PCCPs, nutraceuticals, etc., on human beings. EU, however, took the lead. As usual, EU takes initiative of most of these plastic kind of things. It is there's a hard ban on the use of microplastics or plastics in ingredients and formulations of products from 2027 to 35. So there's a 4 to 12 year transition period, and the transition period is purely due to lack of alternatives or difficulty in formulations. As you'd see, lip, nail, eye care is something that is kept for the last because the alternatives are either very expensive or not available. Mascara is 90% plastics, right? It just has black pigment, black or any pigment, and oils, and the rest is plastic. That's what gives it the texture. Now, those are the things that you want to replace because they come too close in contact with you to begin with. They're not recyclable, and they're just causing nuisance. They come back to us in the form of salts and seafood. That okay. Sorry, can you hear that? Let's just walk you through it this way. Yeah, it's a big market. We all know that. Literally, back the cellulose, right? It's being split out of the bacteria right there. It's an extracellular biopolymer. So one thing about microbes is that when you talk about intracellular products, right, the realm or the boundary of the microbes is the limit of how much you can make. You cannot eat or manufacture more than your size is. One of the reasons why extracellular polymers work out and they're cost effective is because bacteria has to produce it and send it out. So the life cycle is higher and the amount of manufacturing that we do is also higher. This is one of our a patented process where we do non-sterile fermentation. That is, here you don't need to sterilize any of your materials, any of your equipment. This is this beautiful biofilm that the bacteria forms. This is the purest form of cellulose on earth. The yield to field ratio of this to plant is quite funny, actually. If you take a football field and you plant bamboo trees on it, it's going to take you 6 to 8 months to harvest it, and it will give you around zero point 0.5 tons of cellulose, pure cellulose, because the rest will be lignins and hemicellulose, etc. That's not the case with bacterial cellulose. You can harvest it in 7 to 10 days, and it's the purest form of cellulose. You don't have to go through pre processing of lignins, hemicellulose, none of that stuff is available. And also it's biocompatible and edible. When it comes to our innovation, we have three kinds of innovation. One innovation is here where we use the same feedstock and split it into two to manufacture two kinds of biopolymers. We're the only company in our ecosystem that manufactures two biopolymers. Now, why two biopolymers? Bacterial cellulose is a slow fermentation process. PG is a fast fermentation process. Now, what's going to happen is if I only take one polymer, then only one market is going to be available and open to me. But the margins are going to be slimmer. When I take just bacterial cellulose, the margins are even slimmer because the yields are high, but the polymer is too expensive. So, what we do is we manufacture both the biopolymers together, not just so we reduce the capital and the operational cost, but then we can also play around with the blends of the biopolymers and create a new category of product. This also is a patented technique where we blend in bacterial cellulose and PGA. So there's an OH group and COH group that plays and opens up a whole new category of products. Excuse me. So this is our current product portfolio. We are pushing on agriculture quite a bit. So when it comes to structural similarity, PJ, polygamma glutamic acid, has quite actually very close to polyacrylamide structures. So polyacrylamides are something that are used in diapers to hold water, but they're the exact thing that is used in the soils to hold water too. The biggest question that comes with agriculture is how what do you do about food security? How do you feed the world in 2050? We don't have the resources, so we have to definitely work on the yield. What does India do for yield right now? This is the data for Indian agriculture yield. Um, this is the fertilizer response ratio. So from 1970 to 2015, the amount of fertilizer that we've started adding in the soil has significantly increased, but their response rate has decreased also. Meaning, if 10 kgs of fertilizer in 1970 gave you, say, 2 kgs of product, even 20, 30 kg of that product of fertilizers is not going to give you that product. So there's an urgent requirement of not just stubbing your soils with plastics or with fertilizers. You have to add a stimulant as well. In this actually this year no last year Oh, this is sorry. This year summertime, right? There was no rain in Bangalore, and people were asked not to water any of the fields, etc. The biggest enemy of agriculture is evaporation. If I'm a farmer, I go to my fields, I add in 100 liters of water in the morning. By the time it's noon, 70% of it is going to be evaporated. So either it goes groundwater or it just gets evaporated. So the available water for plants is really low. For keeping that high, what farmers were suggested this year during droughts is to put the plastics that are present in your diapers in the soil. Now, imagine the kind of contamination that comes in tubers, the kind of potatoes we must be eating, the amount of microplastics in the potato because it is grown in the soil which is laced with plastics. That's where PJ comes in action. So not only these biopolymers help hold water, but they also give drought tolerance, and they go for they supply better conditions for better microbial community. So here you can see the difference in drought condition versus non-drought condition. You can see that the tissue paper or the germination sheet is still wet as compared to with water, where there is literally all the water got evaporated, hence no germination simply due to lack of moisture and humidity. We have done the trials on French beans and sunflower. You can see that the sunflower grew much faster. The fruiting happened 3 weeks sooner than the general time taken to fruit, and the amount of oils present in the sunflower also higher. So it's a good cash product to have. Here if you see products with bacteria in it, Indian government just allowed biostimulants to be sold with bacteria in it. Now both these biopolymers are made by grass bacteria. So not only that, our it's not that both our polymers are grass, but our bacteria are also grass. They're fit to be eaten. So that again reduces our regulatory issues and time for market to entry. We've done that on carrot. You can clearly see that there's a much better effect while saving water. We're also doing hydroponics. If you like zoom in, you'll see the roots where PJ is added is much, much longer, meaning that there is more humidity and moisture available, also nutrients. So that's about it. Of course, there's competition. Every startup, every microbial startup has a competition. We're not the first ones to do it. We're not going to be the last ones to do it. What we have to definitely make sure that we make it better, more efficient, more cost competitive, and also industrial partnerships. Those are a couple of things that one has to think about when getting into biotech space, especially through microbes, because the customer education curve is quite steep when it comes, when you go tell someone that, hey, here's a microbe and this is what I manufacture. Microbes is something that is still scary to the normal public, right? Yeah, with the gut microbial company, you must know, from India right now, they've explained to people how in the gut, it's important to make sure that your bacteria is happy, there are good bacteria, there's bad bacteria, etc. But when it comes to FMCG and large products, or especially to farming community, where educational curve is a little low, you do have to push the product in a very different aspect. So more often than not, right, what happens is with farmers, we leave out the bacterial story. Now, as much as we love the bacteria and as much as we want to talk about it, sometimes you just have to change the story depending on the consumers. And that's the thing about. So what I mean to say is there's a lot of educational curve required with bacteria yet in India, and we should get there. That's one of our jobs. Just a trivia for you guys, please download this app. It's called Beat the Microbean. It's a app by the European, I mean, Plastic Soup Foundation from Europe. What you have to do is you have to scan your daily hair care, toothpaste, any kind of product that you use, and they'll tell you what kind of plastics are present in your product. What do PCCPs do is, currently they have 1100 or more names for the kind of plastic ingredients they add in the product. So they're making it more and more difficult for common people like us to track what is in our products. And this company or this foundation basically just puts it in a same place, and we can read and understand what we have and do not have. So like label pado movement is going on for food, we definitely are starting a campaign to go for label not only food ka bhi mat pado, but har cheez ka pado, so because there's problem every and anywhere. So that's about it from my side. Now I'm open to a conversation. Let's have a conversation. Ask me anything about microbes. If there's an idea, if there's a business you want to explore, if you want help with regulatory affairs, if you want to do more on this aspect, I'm happy to talk about it.
There are already questions present in chat box.
Okay.
I would like to read them. So the first question is, isn't using PGA causes allergic reaction in some people?
Huh. So polygam alpha PGA, yes, because it's chemically synthesized. But gamma bond, the gamma PGA, gamma polygamma glutamic acid, right? That is the key word here. It is only synthesized, or more efficiently synthesized, only by microbes, and it is not known to cause any allergen to most people yet. But we can't say for sure, because if peanut causes allergy, then so does PGA. So it is a possibility, but we're not looking to go into cosmetics without getting certified. But agriculture and home care is what we're targeting. But yeah, it can happen. So we definitely will put a warning like any other. People are also allergic to retinol sometimes. People are allergic to hyaluronic acid, but we get the right kind of population if you put in the disclaimer. Yeah.
Okay, there is the next question, that is, instead of PGA, why isn't peat moss incorporated?
Peat moss can be incorporated, but what happens is peat moss cannot hold the kind of water PGA does. With PGA, it can hold from 400 to 1000 times its weight in water. And also, when it breaks down naturally by these microbes present, it also gives drought tolerance. Another thing about PGA that I did not mention is its chelation skills. It is a very good chelation agent. So, not only does it help the plant go through drought or salinity or bad pH or stress conditions per se, it also chelates the heavy metals and makes sure that it does not go into the plant. So it's not just water holding capacity, but there's a lot of good benefits that PGA has. In fact, PGA has 80% market share in China. China is the only big manufacturer of PGA. Somehow it hasn't come to India simply because of the costs. How about cost escalation with these products? Sure, the cost is a problem for now. Our entire process was designed to make it so thrifty and so affordable that we currently are the most affordable manufacturers of BNC and PGA. If you go see our process patents, or I mean, they will be published soon enough. For bacterial cellulose, we've completely eliminated the need for what do you call it, autoclaving or sterilization. You don't need a sterile loom, you do not need sterile equipment to keep manufacturing sterile product. Now that reduces your water, your capex, your operational footprint quite significantly. And also, the process by which we've made it non-sterile is also the process where we increase the productivity of the bacteria. So there's a lot of thought when it comes to how do you make, okay, how do you make your bacteria behave like a non-genetically behave like a genetically modified organism without genetically modifying it? You definitely would resort to something like UV radiations or mutagenesis, right? Random mutagenesis. But there's one aspect that you can do, which we did, was directed work directed mutagenesis, so basically adaptive evolution. And this is what helped us reduce the cost quite significantly, and it's the same case for PGA. Hmm.
There's a question then, what about carbon footprint?
Excuse me, sorry. Oh, carbon footprint. We use agro-industrial waste, and also wet agro-industrial waste. Now think about the waste that we chose, right, to manufacture these biopolymers is unavoidable waste. Let's talk about pulping industry. When in season, um, from Karnataka alone, from no, forget Karnataka, one factory in Karnataka alone generates 200 metric tons of mango peels when they are in the pulping season. Now, these mango peels are wet, right? When it comes to dry waste like husk or cereal waste, what you can do is you can easily convert it into biochar or you can convert it into biofuel, etc. But when it comes to wet waste, it becomes really difficult to get rid of. More often than not, these companies or the farmers themselves, with the excess produce that is not sold, they dump it in a land available in their own fields. This causes mosquitoes. The pollution control board is after them, so there's a lot of problem that comes with wet waste. Generally, a company ends up spending around 15 to 25 lakhs, depending on their scale, every month on taking care of these products. So, carbon footprint is something that we're significantly reducing when it comes to using the wet waste. Also, all our factories are co-located, meaning we do not spend a lot of logistics and moving the raw material. Third is we've eliminated the sterilization process, which is also electricity and water intensive. So we've tried to tackle carbon footprint on each front, and currently we're carbon negative to neutral. Hmm.
If participants are having any more questions, they can unmute or they can put it in the chat box.
Yeah, you're welcome. Happy to talk about it.
So if we are not steriling it, like sterilizing them, so how are we getting rid of like harmful microbes?
So there will be no harmful microbes growing because the condition for the broth, the condition of fermentation is so harsh that there will be no other microbe or competitive microbe that can grow in the product. Our product is 99.9% pure. The only thing that we have to worry about is endotoxins from the cell wall of the bacteria, because the bacteria is gram positive and they generally, sorry, gram negative, and they generally have endotoxins in the cell wall. But that's a simple downstream processing that can be taken care of. But we've made the broth condition or the non-sterile condition so stringent that we have a 95 to 97% success rate of non-sterile fermentation. But we also do have downstream processing. So when it comes to wound healing or nutraceutical grade, we do go through a very rigorous downstream processing.
Thanks.
Yeah.
So there is a
API.
question. Yeah.
Yeah.
What is the future of probiotics in India? Like use of lactic acid bacteria as probiotic.
Right. So probiotic, prebiotics are all very standardized kind of inputs, right, in the country or in the world. But the world is now moving to microcosomes, right? Now there's personalized medicines. Now there's, microbes um, given to you for your gut, because everybody's gut microbiota is very different. If you take a study, say in an American gut project, right, the American gut project, the hundreds of thousands of people's guts were sequenced, you'll see that the pattern hunting is very difficult, depending on, do you eat meat or not, do you go out in sunlight or not, how much water do you consume, it's a very personal thing. So probiotics on a very general broad level, Indians already had, because we consume a lot of dahi. But in the future, right, it's going to be more um, probiotics suited to my genetics, probiotics suited to my gut type, probiotics suited to me. That's the future of any kind of smoothies, or any kind of microbial traits that we will be doing. It'll be much more personalized, and it actually, the solutions have been developed, right? Like 23andMe does a quick genomic test for the human genome, like our genome. We do have quick tests for metagenomics or gut genome tests now, and depending on what the kind of imbalance that is present in your gut, you can prescribe the kind of medicines or the foods that gets the gut back to the normal health condition. So yeah, sure. I mean, there's a great future, not just probiotics. Probiotics, especially after you go through surgical treatments, because your gut is quite empty and you've been pumped with antibiotics, so most of the gut is in dysbiosis. You can actually work, and there's a good scope for there. That part, surely. Microsphere encapsulation of API by using biodegradable polymers to achieve sustainable therapeutic effect. Is there, yeah, when it comes to delivery of these products into the gut, when it comes to API, what kind of delivery systems are available? There's skin delivery, there's gut delivery. So when you take a painkiller for your head, right, the medicine doesn't just go to head, it goes to all your body, but also numbs the pain in the head. When it comes to talking about tablets particularly, right, or biodegradable polymers that you're talking about, currently, yes, a lot of plastics can be used the in drug delivery, because plastic is inert. Plastic is not known for sure to cause XYZ problem. It's not linked to perfect problems, right? So plastic is inert, so it is definitely used as a binder. But when it comes to other polymers, what you have to take care is how does the polymer react in acidic conditions, because it directly goes to our gut where the acidic conditions are really, really severe. And as far as your polymer doesn't integrate or disintegrate quite quickly on the way to your gut or in your gut, the polymer should stand very well. So yeah, there will be the effect of polymer on drug delivery is quite significant, and the binders really do matter on the release, slow release of drug, fast release of drug. How do they interact with the polymer? What happens to the remnant polymer? So when it comes to bacterial cellulose, because it's cellulose, it's fiber, you definitely excrete it from your body, and when it comes to other things like PGA, you can also work that out. How many microns of plastics can be used for biodegradable process? I'm sorry, I don't understand. How many you mean the size of plastic particles when it comes to biodegradability? So if that is the question, as I understand it, that is a very country to country based question. Yeah, size of plastic particles. It's a very country to country question. When it came to the kind of scrubs or the beads, right, which are now banned everywhere in the world and slowly coming to India, bead the micro bead problem that used to be there. Whatever sieve size a country has in its water treatment system works out well, especially in some countries like Singapore, where the water cleaning techniques are quite significantly advanced. You can go up to, I would say, up to 500,000 microns on a good day, yeah, in the sieving process or one after the other process. But otherwise, I don't think you can work out or recycle or even stop microbes in micron level. And when it comes to biodegradable process, it really doesn't matter what the beginning size of your product is, right? The product whole in whole should be biodegradable. And another problem that comes with people saying their product is biodegradable does not mean that in conditions of soil, water, air, etc., it can be broken down into the monomeric unit. Yes, it can, anything can be broken down in a monomeric unit. Is the monomer harmful? You've been seeing now lately, right? Research and work has been coming out that some of the biodegradable plastics, very well called biodegradable plastics, release phthalates. Now, phthalates are not nice for the nature or for us. So it is not about what size of plastic biodegrade is useful for biodegradable processes. It's what monomer does it release, what happens with that monomer in the environment. That is more of a question to be solved. Another is novel drug delivery system has a scope with microbes. Hell yeah. If you're talking about live microbes and live activity to manufacture some drugs, absolutely. There have been a lot of data and research done on that aspect. Part of your gut can be engineered and worked out to produce a particular drug at the Q, meaning, so this has been done on the lab scale, but the human trials will definitely take a lot of time because still genetically engineered microbes ke products we cannot use. Imagine putting a genetically engineered microbe in your gut. What if it just stops listening to your cue? What if it goes rampant in the gut, right? So if you're a person who suffers with some certain disease, right? Say something as simple as diabetes, and you need insulin regulation. So if you say put an activator in your gut which starts producing insulin to regulate your body insulin, that is some question that people have solved with microbes on a lab scale. But when it comes to scaling up, it's still a question. The stability, the human interaction of it, there's a lot to do. And how do you control it? How do you make sure that the bacteria doesn't go wild or over produce itself? But yeah, future, hell yeah, definitely. So, any other questions related to microbial work? Yeah, we can use it for animal feed. Absolutely. The product is food grade, and actually it's eaten by us also. Have you had the drink mogu? Yeah, the cubes in it are bacterial nanocellulose. Actually, they are made from bacteria. We just don't know it. And the water holding capacity is 99%, so it looks that bulky, but it's just 0.1% of cellulose in it. So yeah, we do eat a lot of these products on a daily basis. Just unaware. Awesome. So yeah, feel free to contact if you have more questions, if you want to work in this area, if you want to get connected to someone in NTU, more than happy to help. I know microbes are just up and coming, but believe me, there's a lot of scope. Most of biomanufacturing is going to happen in the microbial world. And because they're sustainable, they're very controllable, they're malleable, they're invisible but not invincible. So there's a lot we can do with microbes. So I hope you guys take interest and we see you soon with a company somewhere in Bangalore. That'll be nice.